we have an operational informatics unit that's called Translation Informatics Services, which I've run for the past year and a half. And that's how I got involved in this exciting work that involves center's genetic resources and mining it and making it available for the research community. So this slide shows the objectives for these presentations as they were stated in the announcement. In more detail, we'll start by briefly reviewing CCPM genetic data that we have acquired in the past. We'll quickly go through the idea behind genome-wide association studies and phenome-wide association studies. And then we'll actually dive into the project that leads to the development of the portal, which is a large-scale GWAS analysis using CCPM genetic and clinical data, and design of the portal, which allows easy access to PheWAS and GWAS results and all the implementation challenges that we run into. I hope to spend the first 20 minutes or so going through this, and then the second half will be the exciting part when we actually go to the live demo of the application. And I encourage everybody to think of the gene variant or the phenotype which you are interested in and post those in the chat, and we can query this and see if we can discover anything interesting. And finally, I've put in the chat links to the portal itself and also to the form that we ask you to fill should you be interested in playing with this application yourself. It's a free resource for all CU Anschutz investigators and all cancer center members. So the genetic data, as many of you know, Colorado Center for Personalized Medicine operates Biobank, which consents patients who are receiving care at UC Health facilities to participate in this initiative. Consented patients gets their samples, usually blood, but also saliva, collected, and then a subset of these patients has been genotyped. We have roughly about 200,000 patients consented, about half of them have samples collected, and about 34,000 of those samples actually have been genotyped using a microarray platform, using Illumina mega array. And in cancer research, we used to work with next generation sequencing data, and by genetic data and mutations, we are mostly thinking of somatic variants, which are acquired during tumor development, and we use it in research and for the clinical care. I would like to emphasize that this talk is different. It's about germline variants, those that we inherit from our parents. And microarray technology for typing germline data has its own advantages. Specifically, it allows to look for the variants located in the introns and intergenic regions, which we would not get with RNA-seq or exome sequencing. So this microarray data have been thoroughly QC'd within the Translation Informatics Services. I have photographs of people involved in this work. And we ended up with approximately 1.9 million of variants, which were directly genotyped. And the imputation algorithm have been used to derive approximately 50 million variants using known patterns of inheritance. So we currently have a matrix which has 34,000 patients and 50,000 variants, which was used for all the work that I'm going to show. So conceptually, genome-wide association studies are very simple and are aimed at discovering the association between genetic variants and traits. As many of us are clinicians, diseases would be something that we are interested in. In its simplest form, GWAS is a case-control study when clinical phenotypes are derived from electronic health records retrospectively, and then people with a disease are assigned as cases, and there is also a group of controls which are disease-free, and then each of the 50 million variants is tested for higher or lower frequency in cases versus controls. The result of this GWAS analysis is Manhattan plot, which is shown on the bottom, where on x-axis we have all the variants sorted by coordinate on a chromosome, and on the y-axis is the significance of the associations. As you can see, there is a couple of peaks, which for this particular case signify significant associations between this trait and this variance. So the strengths of the data that we have in the center is not just the genetic data. It's that it's linked to the comprehensive clinical data, which is extracted from the EPIC electronic health record. And that allows us to test as many phenotypes as we can derive from that comprehensive clinical information for many hundreds of diseases. And that's how we got to that idea of running this large-scale GWAS and testing as many diseases as possible. This is the service that I have mentioned, which run this project. I would like also to mention a different way to look at the data, which is called phenome-wide association study. GWAS starts with a disease, so the phenotype, and looks at all possible genetic variants for association. PHIVAS is in the opposite direction. We start with a variant, and we look for all possible diseases which it could be associated with. So, and that large-scale GWAS allows us to do this phenome-wide association study. On this slide, I have shown an example of this PHIVAS analysis. Yesterday, I was interested in this variant on the chromosome 6, which is associated with Graves' disease. I am an endocrinologist, so many of the examples that I will show will be for the endocrine diseases. So, and when I used the portal to run a PHIVAS analysis, I saw that actually the association with Graves' disease is there, but that variant is also linked to many other autoimmune diseases, such as type 1 diabetes. And the strongest association is actually with a celiac disease. So, which I see as kind of a genetic justification of our practice, where if we see one endocrine autoimmune diseases, we start looking for others. The portal will allow to run this analysis for any of those 50 million variants that we have. So there were many challenges and decisions that we had to make to enable this large-scale GWAS analysis. One is to decide how we are going to define the diseases, which is not straightforward. And the easiest way is to use billing codes, ICD-9 and ICD-10 codes. There are obviously many deficiencies of this approach, but there is one big advantage. It's easily automatable. We were looking at many hundreds of diseases. So Laura Wiley's lab have an experience with so-called Phi-code algorithm, which groups those ICD codes into relevant phenotype definitions. And Laura and David were kind enough to share their code with us. We ran that code on our clinical data and were able to define 1,268 phenotypes that had at least 100 cases, which was sufficient to run GWAS analysis. So the next question was how we are going to run GWAS analytically. And one challenges of doing this kind of analysis. It's computationally intense. It's a lot of math. And at that time, we had one algorithm and a tool that's called SAGE operationalized within TIS. And the issue with SAGE, well, it's a wonderful tool, and it was giving us great results when we run one or couple of phenotypes. It was slow. It would take us a long time to run more than 1,000 GWAS analysis. Unfortunately, at that time, Regeneron Genetic Center posted and published another algorithm that's called the Regini, which was specifically designed to be computationally efficient. And this slide shows 2 steps of how Regini works. On the step 1, it's just a regression analysis on a small subset of variants, and then the outcome of prediction for that model is used in step 2, where the actual association test is run for all available variants. And that's a standard approach for the SAGE as well, which also has 2 steps. But Regini has one advantage. It can run multiple phenotypes simultaneously, which is, in addition to the other intelligent design of the algorithm, that's what brings the efficiency. So this is the table from the Regini publication that compared computational performance of SAGE and Regini for 50 GWAS analysis run on the UK Biobank data. SAGE would require about 500,000 of CPU hours, and Regini was about 1/4, would require about 1/4 of that time. So there's a significant reduction in computational requirements. And those results matched our experience as well. So at that time, SAGE took about 18 hours to run on a 60 CPU virtual machine for each phenotype, and Regini took about 4 hours. So about 3-4 times more efficient. We implemented Regini on our high-performance computing infrastructure and ran a series of quality control checks. And one of this was comparing performance of Regini and SAGE. So this is what the scatter plot shows on the right. These dots, they are associations. P-values on an X and Y axis. It's actually negative log 10 p-values. So right upper corner shows most significant variance. And you see that both algorithms produce comparable results with most significant variance approximately at the identity line. We did also compare our results with published ones, specifically from UK Biobank. So left scatter plot compares results for the thyroid cancer in our analysis and UK Biobank analysis, and shows that both projects discovered about the same set of associations. Of course, we have a much smaller number of patients than UK Biobank, which has about 500,000 participants, which is reflected for lower power to discover some associations for common phenotypes, such as hyperlipidemia, which is what is shown in this scatter plot on the right. Sorry. Okay. So we ran Regini on 1,268 phenotypes, which gave us about 62 billion statistical test results in the form of about 5 terabytes of text files. So not the most user-friendly format, really hard to work with, even if you know how to do computer programming. So we realized at that point that to make these results useful for researchers and to be able to extract most useful information out of it, we need a database. We need to put it in some sort of database and ideally put a graphical user interface on top of it, which allows to explore it conveniently. And Chris Genu pointed to the global biobank engine at that time, which has similar functionality, and they were kind enough to publish their code base, and we adopted that code base. The next question was what database to use, and we explored 4 different databases before we ended up on the one that actually worked. So the Mongo database is a NoSQL database. It's free to use, and it's used by an earlier version of the global Biobank portal from Stanford. But ultimately, Stanford folks decided to migrate from Mongo to SciDB because I believe Mongo was simply too small for those large data sets. And a SciDB, that's what currently used by Global Biobank portal. It's very fast, but it requires license and on-premises server, which means not only buying a server, but also having a systems administrator to run it, which we didn't want to invest into this. So, which means we started looking at the cloud databases, which would not require a system administrator. And the first attempt was with a Google Data Store, and I don't know SQL cloud database. We chose it because it natively works with Google App Engine, which is a convenient way to deploy your application online. And we actually make it work for the Kabo portal, which was sort of our dry run, first attempt to make an online application for the GWAS data. And the Kabo portal is currently running at this URL. It's one of the projects, one of the projects of Kathleen Barnes research group. So it worked. The problem is the data store, it's painfully slow to import large amount of data into it. So it worked for the single phenotype for the Kabo portal, which is asthma, but it was not practical for 1,000+ GWASs that we had at the time. So then we started looking at the BigQuery, which is another Google database. It's analytical database, which specifically designed to work with very large data sets. It's convenient, it supports SQL language, and we made it work with a Google App Engine as well. That's what currently is used. So I want to talk briefly about our database schema, because we put a lot of discussions and thinking of how to design it. And that was necessary to make queries fast, meaningful, that has all the data that we need, but also importantly cheap enough that we can make that application sustainable. So currently we have 8 tables in the database. SumStats is the biggest one that stores all those GWAS summary statistics. I'll explain this partitioning by coordinate on the next slide. There are 3 tables with different human genetic constructs, exomes, transcripts, and genes. We run an annotation for all variants in our data set using Ensemble Variant Effect Predictor that gives us information such as RSID, which gene it belongs, where it is located in coding sequence or in the intron, what's a consequence if this is within a coding sequence, and that list can continue. We had a separate database for gene aliases so that the database can be queried by all the names of genes. We added all RSIDs so that RSIDs can be used to query, and top hits is redundant with SumStats, was just added for the efficiency. So BigQuery is a great tool, but it has one big caveat. It's not supporting indexing. For those of you who are familiar with databases, this is what allows an efficient query of the data. Instead, it uses the raw power of Google to just scan through all the database. And unfortunately, it's also charges for all the data that's been scanned. So just a simple raw query of a single variant for our 6 terabyte SumStats would cost about $120, which obviously we would very quickly run out of money if we do that. So fortunately, there is a way to split that big database into chunks. It's called partitioning by integer by Google. And it requires it required a separate, what we call an absolute coordinate for each of the variants. So traditionally, the genetic coordinates is a combination of the chromosome number and the address within that chromosome, which would not work. We needed all this to be integers. So that's why we concatenated all human chromosomes one after another and assigned each coordinate a new address, which is a number between 1 billion and 3.9 billion. And then we split all that coordinate space into 4,000 chunks, so that when we ask for a single variant, it just looks at that chunk, not at the whole database, which reduced the cost of a query for a single variant to less than a cent, from $120. And that's what made it possible for us to offer this application to everybody, because it's relatively cheap to use. So very quickly, there was one other issue. Regini runs multiple phenotypes at the same time, but also it requires that those phenotypes are missing, have the same missingness in the data, which means that those individuals that don't belong to either case or controls are about the same in those phenotypes which are run together. For example, it would be unwise to run breast cancer phenotype, which is predominantly female, and a prostate cancer, which is all males together, because that would give an inaccurate result. So that's why we used hierarchical clustering to analyze missingness in all our 1,268 phenotypes. And this slide just shows one cluster where breast cancer is combined with all the other female cancers, such as cervical and ovarian cancer, which makes sense. And that allows us to get the most accurate result out of Regini. So I think this is the time for the live demo. But before I switch to the live demo, I want to briefly mention who can access this application. And it's a very simple answer. Anybody with CU and shoots.edu email can apply, and we will provide credentials to access it. We actually just had yesterday discussion on the access to the Biobank committee about allowing the other academic investigators from outside non-commercial institutions to use it. And it's now possible, as long as that outside investigator has a sponsor within the University of Colorado. I've posted this link to the user agreement and access request form. Please, if you get interested and excited about the application, go and fill this form. It's very short and easy one, and you will get an email with instructions of how to set up a password and access the portal. So with this, I will stop sharing the PowerPoint and switch to the portal. Okay, well, I hope you are now seeing my browser screen that says Colorado Biobank portal. This is accessible through this URL, which I also posted in the chat. It requires credentials, as I've mentioned, so I'll sign out and then re-sign in. And once your credentials are approved, that's ready for use. There is a single entry point for all the queries. There are a couple of examples shown here in the supported queries box. But basically, if anything that you want to ask query, you put in this window here. And there is a useful feature which auto-completes once you start typing, which helps you to figure out what's the exact spelling for a particular disease that you are interested. So this is a cancer center presentation. So I'll start with some kind of cancer as an example, and we can test the others. So let's try breast cancer. There are a couple of them. One is restricted to female. Let's do just the general breast cancer. The queries are not instant, especially when you query by gene, and that's because the databases are large and the query by gene requires asking for information from multiple databases like annotation and gene structures. So, but it's workable. It takes a couple of seconds to get the result. So right now the slide shows the Manhattan plot for a breast cancer. There is a case and control count here, which is useful to get an idea of how many patients with particular condition we have in our genetic data freeze. There were 1,436 breast cancer patients included in this analysis, 29,000 controls. And this is a Manhattan plot that looks different from what I showed on the slide. That is in part because of the limitations of the web interface. It's difficult to plot 50 million dots in a reasonable time in the cloud. So it only shows 1,000 variants with the lowest p-value, or those that are mostly associated. They have the greatest significance for the associations with the breast cancer. And you can see that there are a couple of peaks in here, which immediately tells you that we were able to discover in this analysis some associations in the GWAS. There is a tooltip which gives you an idea of where that variant is located and what's the p-value. But I think most useful is actually not the Manhattan plot, but that table that follows it, which is what allows you to explore the individual dots, individual variants. So there are a couple of columns. I will describe what these columns show. I think the most useful one, most interesting, is the p-value. By clicking at the column header, it's possible to sort out by the p-value, and we are interested in the variants with the lowest p-value, most significantly associated, and I just sorted by that. And on the first column, it shows the coordinate. It's on a chromosome 16, reference and alternate alleles, and RSID for this variant. Gene symbol, it shows which gene that variant belongs to per VAP annotation. Annotation says where it is located, whether it's in the intron, in intergenic, or within the coding sequence. So allele frequency is interesting. GWAS mostly deals with the common variants. So that number here shows that these variants have high prevalence in the population in about 1/3 or 1/4 patient would have that variant. And the odds ratio shows the direction of the association. So this variant increases the risk of the breast cancer and the degree of it, the effect size. So the consequences, the last column, which I would like to describe, it only works for the variants within the coding sequences. Like here is the frameshift mutation in the BRCA1 gene. And if I sort by this, there's actually a whole bunch of variants within the coding sequences with a various degree of association with the breast cancer. So I'll go back to the p-value sorted view. And yeah, one variant which is obviously caught my attention is in the BRCA1 gene. As we know, the BRCA genes are responsible for hereditary cancer syndrome that increases your risk for breast and ovarian cancer. And this variant is a frame shift, so it's predicted to result in the loss of function for the BRCA gene, and it's likely causative, pathogenic for the cancer-associated syndrome. It also has a very high odds ratio, so it really increases your risk of thyroid cancer, although that number might not be super accurate for a very rare variant like this one has a very, very low allele frequency. So one thing I wanted to mention is that allele frequency is not shown for all the variants, and you might wonder why. And the reason for this is to protect patient privacy. So there are variants which are extremely rare, which might be encountered just in one person or a few persons in the biobank. And our regulatory body, which is an access to the biobank committee, said, no, we don't want you to show any allele frequencies which would correspond to the allele count of 10 or less than 10, because somebody might go into trouble of re-identify these patients, which would be a bad thing. Okay, so this is the view by phenotype, or what I call also a GWAS view. And there are many hyperlinks which allow to conveniently continue to explore if you find anything exciting in this view. For example, it's possible to click on the BRCA gene, so I'm going to do that. As I mentioned, gene queries, they're a little slow because the way the code is designed on the back end, the amount of information that has to move, but I think it's going to work. Yeah. So the gene view has this sort of a graphic representation of a gene where all the different exons are shown on the x-axis, and differently colored dots, those are the variants which are either located within the coding sequences or just nearby. So the splice sites are very close to the coding sequences in the introns. There are a couple of interesting functionalities here. So it's possible to switch between negative log 10 p-value and odds ratio view. I found odds ratio view not that useful. It's possible to zoom in a specific area of the gene, sort of a detailed view, and scroll if there are too many dots. And again, the most useful is a table. Table can be filtered using these buttons to show all variants, just the variants within the coding sequence, and just the variants that are predicted to cause loss of function. So there's a stop gains, frame shift, and splice site mutations. And this view shows that there are a couple of these frame shift mutations, and with a different degree of associations. And here's our friend, which we show on a previous breast cancer GWAS view. So otherwise, the table is very much similar, and it can be sorted by any of the columns. External references are cool, which allows to look at this specific gene in a bunch of databases and even query the PubMed. For BRCA1, I think that's kind of obvious. We'll know, most of us know this gene, but if this is an obscure transcription factor, I find it helpful. It quickly allows you to search, let's say, PubMed, and see if there is an association with your phenotype or that is new. For example, the TOX3 gene, I didn't know what it is, so I looked through the PubMed search and found that it's indeed associated. It's a transcription factor that facilitates estrogen receptor signaling, and the variants in this gene naturally associated with the high risk of breast cancer. So the next view, which I think is interesting to look at, is a FIWAS view, and that can be accessible by clicking at the variant. Sorry for that. Okay, so I clicked at this variance, and now I have this FIWAS view where on the x-axis we have all 1268 phenotypes. There's a tooltip functionality which allows you to see which phenotype specifically each bar represents. So the tallest bar is obviously breast cancer, but there are two others. So if you're looking at this, the second is breast cancer just restricted to females. That makes sense. And the third phenotype is malignant neoplasma in female breast. So kind of not the most exciting FIVAS plot in this case, but yeah, it just shows that the results that underline this analysis are making sense. So to sort of demonstrate a more meaningful FIVAS analysis, I'll use the thyroid cancer phenotype, which is my favorite. I'm an endocrinologist specializing in the management of thyroid cancer. So this is the GWAS plot that shows a couple of peaks in the Manhattan plot, which are significant associations. So I'll sort this by p-value and click on the top hit, which is located in PTC-SC2 gene, which stands for papillary thyroid cancer susceptibility gene 2. So and I'm getting emails, which means some of you are signing up for the portal. So I appreciate the interest.
I did paste the link in the chat, Nikita.
Yeah, I did that too. Okay, yeah, so this is the FIVAS plot for the most significant variant associated with thyroid cancer. And when I started looking at the other associations, hypothyroidism came up as the top hit. And interestingly, the odds ratio is less than 1, which means I would think if you have a thyroid cancer, which is treated by thyroidectomy, which makes you permanently hypothyroid, the associations would be in the same direction. But it seems like those variants, which makes you more likely to develop thyroid cancer, make you less likely to develop hypothyroidism, which is a clinically relevant information, which we are actually exploring in collaboration with surgery resident Martin Barrio. And our very preliminary data showed that if you have Hashimoto thyroiditis, which is a most common cause of hypothyroidism, then you actually are less likely to have a most aggressive form of thyroid cancer with high probability of recurrence. So it's just an example of how that kind of analysis could be useful. Well, I think I'm getting close to finishing the demo. There are a couple of useful resources which I would like to mention. In the FAQ tab, there are guides on how to use this portal if you have additional questions. Those guides were put together by Fang, and the credits is my acknowledgement slide. So the informatics is a team sport, and the projects like this involves many people, which are all listed here. And my special thanks to everybody who took in different aspects of this, such as project management and cloud infrastructure, high performance computing, and GWAS analysis itself, and also to the global Biobank engine folks whom we took the very first version of the code, which we had to modify extensively since. So there are many messages in the chat. So I'll yeah.
I was trying to answer some along the way, Nikita. We definitely have time for some verbal questions.
I just
Yeah.
asked what's meant by region on the previous slide.
Okay, yeah, so not all variants have an assigned gene in the VAP annotation. So to be able to query variants which are in these intergenic regions, we have implemented this search by region, which you format by having a chromosome followed by 2 coordinates, the starting and the end of the range. And I think it limits the size of that region to either 50 or 100,000 base pairs. So yeah, that's the idea behind it.
Thank you.
And I saw Russell Bowler put a couple of RSIDs. So I queried one on the chromosome 4. There are weak associations for this variant. So and seems like the strongest one is a protective against diseases of hair and hair follicles. So I don't know how meaningful that result is. We can try a second one.
Hi, Nikita. Sorry, this is Martha. I have a question. In terms of allele frequency, you probably consider, for instance, in that case, what is the frequency of the C versus the G? But is there any way to find information about heads versus homozygous? Because sometimes, primarily if it's a recessive, you may need an homozygous for the phenotype. And I don't know if that's something that can be found also in the database.
Yeah, so what is shown in the portal is a minor allele frequency. So that's a frequency of allele, not a genotype.
Yep.
I don't think you can explore genotypes directly in the portal at this time, but we can pull that data from our database just on the back end. If you submit a request to TIS, Translation Informatics Services. But yeah, no automated feature like this at this time.
Okay.
Can I ask the question while the camera's on us? So first off, thanks so much for doing that, Nikita. It was a great tutorial. I think it's really straightforward to use it. So obviously you're limited, by your power, depending on the frequency of the disease, the frequency of the allele, the odds ratio, and so forth. And so, how are you going to combine with other databases, or how are you going to expand this to, in order to be able to increase that power so someone could ask a question, particularly if we wanted to ask things about, rare diseases, for example?
So that's obviously a limitation. I mentioned it on the hyperlipidemia slide. We only have 34,000, which sounds like a large number, but in a population genetic scale, that's a relatively small database. It's expected to grow. So 2022 is an exciting year. We will likely get about 40,000 exomes from our collaboration with Regeneron Genetics Center. And there will be also a bunch of samples genotyped using microarray technology. And once that data, we have that data and process it through the QC pipeline, we hope to incorporate it in this portal. So that will improve the power, at least twofold, maybe more.
Are there academic efforts that, other hospitals doing something very similar to what is being done here?
Yeah, that question actually probably will be better answered by Chris Ginu, who is involved in some of these collaborations.
Yeah, I mean, we're building. So one of the things that we're trying to do here is sort of set up an infrastructure where we can think about this stuff across health systems. and still, within our regulatory framework. So in particular, we work a ton with Mount Sinai and UCLA. And they have similar sized sort of repositories to ours. So a lot of this work kind of happens through that framework. And then we've got sort of larger initiatives for meta-analyses and sort of trait-focused consortia and stuff that we work with in a lot of different contexts.
Great.
So
Great, thanks.
Yeah, and I think at this time, we kind of we've exceeded the functionality of the mothership, which is the global biobank engine. This is the most functional portal of sorts, which I know about, and we are absolutely open to collaborations where we would either port the code base to the other biobanks, or we would host the other data within our portal.
Yeah. Great, thanks.
Yep. Just the result for the other RSID showed that it's actually has significant association at the genome-wide significance level with rheumatoid arthritis. The second RSID, which was posted in the chat.
Thank you.
Okay. Yeah, I think I've answered the region question. So there's no any specific meaning behind that, except this is just a bunch of nucleotides together. There's a specific coordinate. Batch functionality for submissions. No, there's no batch functionality this time. Query by ancestry, not yet. Again, we are working with Chris on starting adding some of the ancestry information to the portal, which we might have some prototyping done in February. That's a future work. So at this time, you cannot subset GWAS analysis by ancestry. And frankly speaking, we don't have enough numbers for most of the phenotypes to run ancestry-specific GWAS. But with a larger database in 2022, we might.
I'm sorry, Nikita, it's Russ. I didn't catch your response to the batch queries.
No batch queries. If you want some large scale, lots of information, don't try to scrape it of the browser. Just reach to us, reach to Translation Informatics Services. We have pipelines where we help researchers to get large scale data from the biobank. The portal is mostly for exploration, hypothesis generation type of work. So no batch functionality for the portal, but there is a batch functionality on the TIS side. Can we link any of this data to patients in Epic? So the portal is a low security application. We are not allowed to store any line level data. It's all summary statistics. We obviously have line level phenotype data for all the biobank individuals within our unit, which can be queried if necessary.
So I'm just trying to understand how best I can use this. So if like I wanted to see if a particular genetic variant was associated with a response to like a treatment, for example, how would I be able to go about doing that? Or is that even possible at this time?
Yeah, it is possible. So the traits that we have in the portal, they are derived from the billing codes. So they may not be a phenotype specific to your treatment that you are interested in. There is a way to define this phenotype as a custom one and just run a GWAS and potentially add it to the portal. The process for this is going through the access to the Biobank committee. So, and all the links are available if you Google Translation Informatics Services. So I probably can do it. TIS. No. We have a portal, the website.
Yeah, I
Okay.
see this translational informatics, CCTSI.
It's not CCTSI, it's CCPM.
Oh,
Lots of
CCPM.
similar letters. Yeah, I'm not sure what I'm sharing, but if I'm still on my browser, you should be able to see the website for TIS.
Yeah, I can see
yeah.
it. Thank you.
So there is genetic data delivery services and biobank exploration services. So if you submit a request here, then depending on the type of request, we might be able to help. It might involve health data compass if that requires a detailed phenotyping work, which we are only equipped to do to a certain extent in TIS, but we have the ability to run GWAS on any phenotype.
Great, thank you.
And Nikita, do you have an estimate of when I know Regeneron's planning on getting up to 250,000 or something like that, when those data might be available?
Yeah, 250,000, I don't know. It's at a smaller scale right now. I think we have about 50,000 samples submitted to them at this time, and they are promising. The latest promise is in January, so they have 5 days to fulfill their promise.
Okay. So it's going in batches, like 50,000 or something like that, or yeah, okay.
Yeah, Christy Crooks, who runs the Biobank lab, sends those batches to Regeneron, and they lump them together and they put it on their sequencers. And we hope to get the first batch very soon. Okay, so we still have 5 minutes. I'll try to answer as many questions as possible.
There's a question in the chat about for missense and LOF mutations. How are you determining if the mutation actually affects protein function? And is that factored into the odds ratio and p-value?
So the second half of that question, no, it's not factored in. The predictions are done by the web tool, and it's an ensemble annotation tool, variant effect predictor. So it's automated. Yeah, and the link to the portal has been posted by many of us. And I think these are all the questions. If I missed something, please ask. I'll try to answer.
Cool. Well, thanks so much, Nikita, for this wonderful presentation and for all your hard work in developing this tool. It's really an awesome resource for all of campus.
Yeah, it's my pleasure. And the email has been clicking and clicking and clicking, which means we got quite a few users, and that's great. We want this to be used as much as possible. That justifies our effort.
Agreed. Cool. Well, thanks everyone for attending.
Yeah.
You're popular today, Nikita. There were like 75 people on the call.
Yes, that's exciting.
Thank you. That was
you.
great. Yeah, thanks.
Thank you. Goodbye.
Bye, everyone.
Thank you, Chris. Thank you.